Friends, welcome to Time with Thebsian on medical devices. Today's topic is testing of biomaterials according to United States Pharmacopeia. United States Pharmacopeia is the Bible for testing of drugs as well as medical devices. We started designing tests for plastic containers for keeping medicines. But as years passed by, medical devices flooded the market and so many regulations have been set up, so many testing methods have been developed, but the USP test for biomedical devices or implantable devices or any medical device that is going to be used in human beings or animals, USP testing methods stand alone as a solid testing method. Today we will learn about USP testing methods. This episode was added because there were a lot of questions regarding the earlier episode on classification of medical devices. Many told that it is so congested or confusing, or it was not simple to understand for the common people. That is why I have added two more episodes, one on USP testing of various classes of medical devices, another one testing of biomedical devices as per United States FDA, Food and Drug Administration. So today we will see what the USP test methods are. And dear friends, if you have any questions or doubts, kindly you have the freedom to ask me, telephone me or email me. USP test method is the most common test method to determine the biocompatibility of medical devices, whether it is a simple syringe or a plastic tubing or an implantable medical device like a heart valve or a pacemaker. USP also has a companion called National Formulary. These two are considered to be standard texts for testing drugs as well as for testing medical device. As per USP, there are 6 classes of testing. Class 1 is a simple testing method. Class 2 addresses materials with little risk. Class 3 addresses materials with little more moderate risk for humans. Class 4 and 5 addresses tests for materials or devices that may considered to be more risky when applied, when used in human beings. So more serious tests are included so that class 4 and 5 can be sanctioned for that particular devices. Class 6 is the most stringent class in USP methods. the device has to pass class 1 test, class 2 test, class 3 test, class 4, class 5 and class 6 test. The most critical device has to pass all 6 classes of tests as per USP testing methods. Why people go for USP testing, class 6 testing? Once your product is tested and meets all the requirement of class 1 to class 6 of USP tests, it can get a certificate for final use, or it can get a certificate to go to the marketing of the device. That means tubings, catheters, single-use items like that, and permanent items like pacemakers, heart valves, stents, etc. definitely go for USP tests, simply because it gives confidence for the manufacturer and the seller and the end user, that is the doctor who prescribes a medical device that has passed the USP tests. They all get the assurance that their product are top ones in the market, and they have the safety tests done so that they are biocompatible as far as the human regulations are considered. See, in the case of class 1 tests, the test material is prepared as per the USP instructions, and they are extracted in 0.9% saline for injection, and these extracts will be prepared in an oven at 70 degrees or 121 degrees, sometimes for 24 hours, sometimes for 48 hours, sometimes for 72 hours, depending upon the decision of the toxicologist who is going to carry out the testing, because he has got all the details regarding the nature of the plastic material or the test material, what temperature it can withstand, and what is the end use of that material. Depending upon these 3 factors, the toxicologist decides how and for long it should be extracted, at what temperature, to test, to carry out the test for class 1. And the extracts are cooled down and injected into laboratory mice at a dose of 50 ml per kg body weight. 0.2 ml of the same extract is injected intradermally on the dorsal side of a rabbit. These 2 tests are carried out by the toxicologist, and the animals are observed for any kind of untoward reaction for 24, 72, and how many hours the toxicologist will decide. If the product or the test material has to pass class 2 test, they will be extracted in 1 in 20% solution of alcohol and 0.9% sodium chloride injection mixture. The injection will be carried out as mentioned for the class 1 devices at a dose of 50 ml per kg in the laboratory mice, that is intravenously, most likely through the tail vein, and 0.2 ml at 5 sites on the dorsal side of a rabbit and observed for 24, 48, 72 or the time periods decided by the toxicologist. For class 3 devices, along with class 1 and class 2 injections in mice and rabbits, the test material is also extracted in polyethylene glycol 400 and the extract is injected as per the dose mentioned for class 1, class 2 testing devices in mice and in rabbits. They are injected not intravenously but intraperitoneally in mice and intracutaneously in rabbits, the same way as for class 1 and class 2 tests. The toxicologist records his observation for untoward effects. For class 4 certificate, the product test material will be extracted using saline, 1%, 20% in saline and polyethylene glycol 400 and tested as per requirement for class 1, class 2 and class 3. But as far as the class 4, another test is also included. Test samples cut into 1 into 1 into 10 mm size will be implanted on 5 sites on the paravertebral muscle of rabbits. The number of rabbits and the time of implantation of period for observation will be decided by the toxicologist as decided by the USP test method. After the desired time, maybe 72 hours or 5 days, the implanted site, implanted materials along with the surrounding tissue will be excised after euthanizing the rabbit, and the toxicologist will cut the implanted site to see how the response is. He also may send this tissue to the pathologist for histopathological observations. There is another alternative for this rabbit implantation test. Test strips could be implanted subcutaneously in rats and also observed after the desired period of test time. For the class 5 materials, the implantation test is excluded and all other tests for class 1, class 2, class 3 and class 4 are carried out. For a product to pass all the class 6 tests, the material should undergo testing indicated for class 1, class 2, class 3, class 4, class 5 and class 6 also. And you can see that in the class 6, the implantation test is included and the time period is decided by the toxicologist as required by the USP test. If once the product, the test material passes all 6 tests described as per USP testing, the material is passed for biocompatibility. The product could be certified for biocompatibility. It can be called a biocompatible material if it passes all the 6 tests in the USP. So therefore, we can see that even when the manufacturer subjects his products for other tests indicated by FDA or European testing or Japanese testing, they also make sure that their products are tested for all USP 6 tests. Friends, I hope this time you are more clear about USP tests. And USP tests in general are carried out using different types of extracts for extracting the test material. So when the extraction is carried out in different extraction media, the possibility of ingredients or components of composition of the test material is increasing. It will be more than that of the test material is extracted in saline. The test material is extracted in 1 in 20 of alcohol mixture. Then the extract is polyethylene glycol 400. And depending upon the nature of use of the material, the time of extraction and the temperature at which the materials are extracted will be varying. 70, 121, like that. 70 degrees and 121 degree centigrade. So, then there is an implantation test in rabbits and in rats. One more thing I will mention is, when these injections are carried out, the extraction media are without testing material in the extraction oven, and those extracts are injected in another set of control animals so that the difference can be observed. So like in implantation also, there are negative controls. The materials which when are implanted on the contralateral side of the test material, the negative controls will not show any kind of tissue response. So it is very easy for the pathologist to compare the response of the test material to the response of the negative control. So these are the technical parameters which I explained later. So as a common citizen or an ordinary person, these episodes give more assurance that when a medical device is tested and certified, they come into the market as safe products. So friends, we are winding up this session. Kindly let me know your doubts and questions, and I welcome you people for the next video next Friday. Kindly let your friends know about this series of videos. Kindly like and share these videos when you watch them. Thank you very much. See you next week.